Hi, IB students. Today we're going to continue with 8.2 and we're going to focus on the structure of the mitochondria. So that happens to be the essential understanding here. So the understanding is that the structure of the mitochondria is adapted to the function it performs. So think of what you learned back in the 9th grade, that form fits function. So what are the mitochondria? These are the power plants of the cell, right? The powerhouse of the cell. So they synthesize large amount of ATP via aerobic respiration or cellular respiration, as we're starting to study. All eukaryotic cells possess mitochondria. Those aerobic prokaryotes that do not have mitochondria, they use cell membrane to perform the oxidative phosphorylation process that we see happening within the inner membrane of the mitochondria. Now, we know through the endosymbiotic theory that mitochondria, they are thought to have once been independent prokaryotes that were internalized by eukaryotes. And that's what we know as endosymbiosis, right? When a bigger prokaryote engulfs a smaller prokaryote, and then it becomes a eukaryote, and then that eukaryote will engulf another smaller prokaryote. So what do we need to know about the structure of the mitochondria? So a couple of key features here is that they have a double membrane, and all of these features agree with the endosymbiotic theory. They have their own DNA that's circular and naked, and they have their own ribosomes, 70S ribosomes. And their metabolic processes are susceptible to antibiotics. So all of these key features here suggest that they were once free-living independent prokaryotic cells. All right, so what must you know about the structure and function of the mitochondria? So this is what the mitochondria diagram looks like. So let's start with the outer membrane. So the outer membrane contains proteins for shuttling pyruvate into the mitochondria. So the process of glycolysis, which is one of the 5 stages of cellular respiration, this is going to happen outside of the mitochondria in the cytoplasm of the cell. So one of the key features of glycolysis is that it's able to make pyruvate. So that pyruvate molecule needs to be shuttled inside the mitochondria. So the outer membrane of the mitochondria contains proteins to be able to shuttle in that pyruvate to the inside. Now let's look at the inner membrane. The inner membrane right here contains the electron transfer chain and ATP synthase for oxidative phosphorylation. So that means that the electron transfer chain and the process of chemiosmosis, they are both going to happen within the inner membrane space. And this is what allows for oxidative phosphorylation, which means that the majority of ATP is going to be produced in this process right here in the inner membrane. Now let's look at the cristae. So the cristae is the highly folded inner membrane pockets, to say, and the inner membrane is folded to increase surface area to volume ratio. So there's more available surface for these processes to occur. And then we have the intermembrane space. So we're talking about the space in between the outer and the inner membrane. So the intermembrane space is small, and that's important because that means that that small space is going to maximize the hydrogen gradient upon proton accumulation. So a hydrogen gradient needs to build inside the intermembrane space for ATP synthase to work properly. And you'll see that later when we look at these processes in more detail. And then we have the matrix of the mitochondria, and this is the interior of the mitochondria, or the central cavity. And it has enzymes in there, and it has a suitable pH, and it's all appropriate for the Krebs cycle, or the citric acid cycle that is going to happen inside. So here we're starting to learn a little bit about some of the processes within cellular respiration, right? Glycolysis, which happens outside of the mitochondria, the Krebs cycle, which happens in the matrix of the mitochondria, and the electron transfer chain and chemiosmosis, which happens in the inner membrane space of the mitochondria. So definitely form fits function as far as the mitochondria is concerned. All right, so one of the skills for this section is for you to be able to annotate a diagram of the mitochondria. So you can definitely use the information that we just talked about just now to indicate the adaptations of the mitochondria to fit its for its form to fit its function. So they might give you a diagram that looks like this, which is an electron micrograph, or they might give you a diagram that is just a representation of the mitochondria and it looks like this. Now, don't get confused when you look at your mitochondria because, let me click here. Okay, we got labels. Because it might look a little bit different depending at what angle the picture with the transmission electron microscope was taken. So it's usually sausage shaped. We usually think of a bean shaped sausage shaped structure. And then we can see the internal protrusions here, or the foldings of the inner membrane. So all of these internal protrusions here, that's the cristae. And then we see them here as well. And then we see the intermembrane space that is also very small. So that's the space right here between the outer and the inner membrane. And the intermembrane space, again, it's colored here in pink. And we don't really see ribosomes in mitochondrial DNA. It's just not visible at the resolution. Okay, so how do we prepare these electron micrograph pictures? So you started recognizing these micrographs last year. So let's learn a little bit of how these pictures are done. So the application is to understand how these pictures are taken through electron tomography. And this produces the active images of the mitochondria. So what is electron tomography? So electron tomography is a technique by which a three-dimensional internal structure of a sample can be modeled. So that's the definition of electron tomography. So samples are imaged using a transmission electron microscope, and then there's many images that are taken. After each image, the sample is tilted to a different angle relative to the electron beam. The images are then compiled and they are used to computationally reconstruct a 3D representation called a tomogram. So you take many pictures and you reconstruct it into a three-dimensional model. When you're dealing with biological materials such as this, you first have to prepare your sample. So in order to prepare your sample, you either fix it and dehydrate it, or fix it, and that's what we call cryogenics. So that doing this is going to stabilize the biological structures and prevent any water from expanding or distorting the image. So based on electron tomography, we're able to get these micrographs of the mitochondria, and we're able to see the different structures of the mitochondria, right? We're able to see that the cristae is continuous within the internal mitochondrial membrane. We're able to see that the intermembrane space is very small and it's consistent throughout the entire mitochondrion. Mitochondrion is singular, mitochondria is plural, by the way. And the relative shape, position, and volume of the cristae can change in active mitochondria. So again, if you see sometimes mitochondria in a micrograph can look circular, and that is again because of the position or the angle where the picture was taken and the 3D image was recreated. So don't be fooled by that, because this can be a mitochondria, but this can be a mitochondria as well. Okay? Okay, that's it for now.